ciprofloxacin. This is the presentation outline. I'm going to take you all the way from background to acknowledgement. Now, South Africa and the world at large are facing a critical challenge related to water crisis owing to rapid growth in population and industrialization. Each day, there are a variety or a number of chemicals that are being discharged into water system, which can pose a severe problem to our health. Nowadays, there are new classes of contaminants that have been a topical issue of concern, which are pharmaceutical products. We may all remember that around 2 years ago or 3 years ago, the country or the world at large, we were facing a crisis of COVID-19. When the COVID-19, because there was no known measure that were there to treat COVID-19. However, some of the antibiotics were being applied to just reduce the impact of the virus. Owing to that, the number of these pharmaceuticals, they are frequently being detected in water bodies. And I will consider the wastewater treatment facility as the hotspot of this problem, because all these pharmaceutical products, they get discharged from a pharmaceutical industries. They went through to the wastewater treatment, maybe from a household, from animal. They go through to the wastewater treatment plants. But now we may all remember that this treatment facility, when they were designed without the intention of treating such compounds. Now, because also the fact that these particular contaminants, they appear in a very, very, very low concentration, of which some of these factor like growth in population, this facility, they end up being the capacity that they receive. Now it is more than they can handle, so which in turn, these products, they end up in our waterway, which can pose a detrimental problem to our health. Of course, there are a number of measures or number of techniques that have been reported to can monitor such contaminants. However, all these techniques, they are sensitive towards this, the monitoring of these pharmaceutical compound. But the problem with these techniques is owing to their cost, or they're limited to high cost. They are very complex in terms of operation. They require someone who has skill to operate, which make them not suitable use for particular measures. However, electrochemical sensing technologies have been reported to be the most common or most recently used technologies due to their rapidness, due to their highly sensitive, and they are also affordable, they are portable. Electrochemical tools, they usually use a sense of an electrode. You have an electrode, and that electrode has also, it's also a problem with regard to the conductive nature. For this electrochemical tool, we need a suitable type of materials that we can cast them on the surface so that they can promote the movement of, of electron. So now, this electrochemical system, remember, its mechanism is, it's, you have your electrode, you have your, what can I say, your interface, let's just say in this case, you have your water system that you want to analyze. What happens is you get your response as a current, and this current is proportional to the concentration that is present in that, environment. Here in this study aims to develop an electrochemical sensing tools for cyprofloxacin detection. Of course, because when you have an electrochemical system, you have to prepare a certain materials that would be suitable for particular application that you are targeting. You will have to prepare the materials. After you prepare the material, you'll have also to understand or interrogate if whether you have managed to come up with the materials that you are interested on. From after that, you then, when you are satisfied with the materials that you have prepared, then you went on to develop your platform and towards an application. However, for the sake of this presentation, I will only focus from the objective to number 4. As I mentioned, once you have prepared your material, there are some certain techniques that you need to use to interrogate the characteristic property of your material. And the fact that also when you prepare materials, you have to prepare the materials that are environmental friendly, that are affordable, because we are running away from using highly expensive instruments to an affordable technologies for this sort of system. In this case, we use an FTIR to interrogate the functional group that are present within our materials. We also use a Raman and also a XRD in order to understand the phases and also of the materials. And then once you understand these characteristic properties of your material and you are satisfied, you then also go to interrogate your material in a sense of electrochemical characterization. This is where you go on by optimizing the materials that you have prepared at a certain interface. In this case, we used ferrocyanide as an interface or as an electrolyte. Here we want to understand the nature of the movement, the mobility of electron within the system. And when we interrogate the system, we realize that when we don't have the material within so this pointer, sorry, it isn't really pointing there. So when we interrogate on the bare electrode without your material, you can see we don't have a high current response, because now the bare electron, the bare electrode, the electrode without the material, it is not that conductive. Hence, that's why we prepare material to improve its conductivity so that we get highly electron movement. But it gets to our attention that when we improve the material, when we put the material, we had an increase in the current response. Also, we further interrogate that material in a terms of charge transfer with using the EIS. And so we also saw when we don't have the materials, we don't get we have a high diameter of a semicycle, which is proportional to the resistance within the surface. But now when you have your material, here probably I'll be focusing on the composite. When you have the composite, you have a small diameter of a semicircle, which then tells you that now you have improved the conductiveness of your electrode, which promote the movement of electrons. After we interrogate them, we then, just like any technologies, say for example, when they're designing a car, before they launch a car, there are some parameters that they go to optimize to understand the potential applicability of that certain device. So at this stage, we wanted to optimize pH, because we know our environment or water bodies, there's a pH is a factor with regard to also the compound that you are targeting. And we optimize the pH from 3 to 9, and we could see that as we are increasing the pH, also the currents respond were decreasing. And in this case, we only chose the pH 5 as our optimum. We also wanted to understand the kinetics of the system. So let's say, for example, like I put example of a car. Like when you have a car, if you are driving at a higher speed, chances are the car is not stable. Also, electrochemical in terms of electron, we don't want the scan rate to be high or to be low. We want the scan rate to be at a suitable level. And we then optimize the scan rate from 1 millivolts to 100, and we realize that as we increase the scan rate, there's also a potential shift in the peak current potential towards the positive direction. And then a proof of concept, we then wanted to understand what happens if we are increasing concentration of a compound of interest within the system. And it came to our attention that when we increase the concentration, there was a rapid increase in the current, which is proportional to the concentration. However, the interest part is that this technology, it goes down to a very, 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 very low concentration, of which some of the technique that have been there, they cannot go, they cannot detect, which is something that is what we are focusing on. We also understand that in the water bodies, there are number of compounds. So you don't want your system to be affected by the environment. You want your system to be targeting a specific group of analytes that you are targeting. We managed to optimize the effect of interfering species, and we found that our devices were selective towards our analyte of interest. We also optimized the nature of the stability in terms of once you use this thing, for how long can this thing be stable? And we saw that after 12 days, we have some stability, but from first day to 12th day, there was some decrease in current response. This is a conclusion. The sensor, it exhibited a very low detection limit of about 0.003, and also wide linear range. And we did application in real water system, and we found the nice recoveries of about 92 to 104. These are some of my references. I would like to acknowledge the following people who have been there for this part of the project. And also, I would like to acknowledge the ASA for giving me this opportunity to be here, and you for listening to me. Thank you.
All right, thank you so much. I want to open the floor. I got only 1 minute for him. Anyone who have a burning question? Yes, there's a question.
What is the impact of temperature and pressure on the current that you find in your electrolyte? Thank you.
What is
how does changing pressure as well as temperature affect your electrochemical?
Okay, so I don't know about the pressure part, but with respect to the temperature doesn't really have an effect. There are some results that we characterize for our material, because remember, this system, it is basically controlled by the material that you've prepared. So you want your material to be stable in any environment. So you want your material, you don't want the material that is not stable at this particular environment and is stable. You want your material to be stable because remember this device, you want to apply them in a harsh environment. If we are talking about wastewater environment, I refer it as a harsh because there are many factors that occurs there. So if the material is stable, it's not affected. Thermally, it's stable, so it doesn't really affect. But pressure, I'm not sure because we have not done the study on that. But I hope in future it can help. Thank you.
All right, thanks to everyone. Can you just give me a round of applause? Thanks, Colin. All right, our next person online is P. Nwanza. Are you here? Oh, okay. Once you're done, for the second presenter. Go.
The first one.
Was he? He's good.
Colin, yes.
Yeah. And now?
Now we're here.
No.
Yeah. All right, the floor is yours.
All right, thank you very much. I want to start by asking you a question. Have you ever wondered when you sit on the toilet, go and do your business, what's actually happening to that excrement? I know it's one of those topics you don't really think about when you're sitting on the toilet, right? But that's actually a very important topic to me and to my colleagues. So I'm asking the question, what happens when you flush the toilet? Where does that excrement go? What happens to it? Could there be any valuable information in that excrement? Perhaps not for you, but definitely for me. So I'll be waiting on the other end for that excrement to get to the other side. I'm Patrick Nwanza, and I'm going to talk to you about what we're doing in the wastewater surveillance group to advance public health in the Nelson Mandela Bay. So wastewater-based epidemiology gave us a platform to survey SARS-CoV-2 presence during the COVID-19 pandemic. And based on that, it has become an important strategy or element of the public health system in order to combat various diseases, not only COVID-19. This method can actually be used to monitor and track concentration levels of various pathogens that are found in water, as well as to monitor water safety, as well as pandemic preparedness. Because if one thing is for certain, COVID-19 hit us when we were not prepared at all. Wastewater surveillance is also very critical because there are members of the community that just do not have access to healthcare facilities, or will get to the facilities when it is too late, when with some of these diseases they are notifiable and they need to be communicated as soon as they have been identified. So this is one method of making sure that the healthcare practitioners are aware of what is happening in the community. So what happens is that your microorganisms, whether they be bacteria, they be viruses, their genetic material is shared in human feces, is shared in saliva, in sputum, in mucus, and it ends up in wastewater. Right? You can imagine yourself, if you've got a respiratory infection, you will cough up sometimes. It's not every time that you're going to spit, sometimes you swallow it. Nasty, but yes, you do. And it goes into your respiratory, into your digestive tract, and obviously ends up in wastewater. So with wastewater, it allows us to track these pathogens and monitor them at the community level. This has various advantages. So for example, when we are clinically testing individuals, it's one person at a time, but with wastewater sampling, it gives us pooled samples, so that are representative of the entire community. Wastewater surveillance also serves as an early detection system for infection. So before it is picked up in the hospital setup, it can actually be picked up in the wastewater. And this is also very important, again, when it comes to pandemic and epidemic preparedness. It can be used also to identify new variants before they are picked up in clinical samples, as well as to predict the trajectory of an outbreak or a pandemic. So whether we are heading towards a peak in infections or a decrease in infections. That, again, can be determined by wastewater surveillance. I'm going to give this overview of the methodology for wastewater surveillance. So once again, all right, so once again, you have your samples coming from human excreta and what have you, and that all ends up in your sewage systems, all right? And from your sewage systems, this is where now we come in, we collect samples, we take them to the lab. As soon as we receive the samples, we extract RNA and DNA, all right, depending on the type of pathogen that we are looking for. From there, we then amplify the genetic material using quantitative PCR or conventional PCR. If need be, we can also then sequence those samples, especially when it comes to variant detection. From there, once we've either detected or quantified, we then obviously have to report to the public health authorities so that the circle is complete. I'm now going to give a snapshot of some of the pathogens that we are looking at in our lab. The first one that I'm going to look at is the SARS-CoV-2, which is where most of this work has actually come from. And in this case, I'm just taking a snapshot of some of the tracking that we have done around a major sporting event. We take samples every week almost without any fail, and we also report this to the university as well as to the Medical Research Council as well as to the Nelson Mandela Bay municipality. So we can see here that in this particular instance, there was a major global sporting event that brought thousands and thousands of fans and tourists to the Nelson Mandela Bay. This is from 2022. And we can see that normally with this kind of event, tourists start to arrive a week before the actual event, all right, and they will probably leave soon after the event. So we can see infections were quite on the, or rather, the copy numbers were quite low at the beginning, and the actual tournament was around about the 1st of April, but we then see an increase in the copy numbers about a week after the tournament was finished. And this is actually in line with what epidemiologists say in terms of the progression and in terms of the detection of SARS-CoV-2. We also observed a similar trend in the data that we looked at for 2023 around, again, the same sporting event. So again, here we can use this information to communicate with municipal authorities, to be aware when having such large gatherings and such sporting events, to be aware of the potential of such spikes in the number of SARS-CoV-2 infections. Another microorganism we've been monitoring is Vibrio cholerae. You will know Vibrio cholerae is the bacteria that causes cholera. Obviously, cholera is an acute diarrheal disease, and it leads to severe dehydration and can kill within hours if it is not treated. So obviously, last year we had that major outbreak that took place in Gauteng, and that prompted us to also start to monitor our water systems to check whether this was actually present in the city. So what we did was, once we received our samples from the municipality, we left them in alkaline peptone water, which was basically to enrich the samples, and then we inoculated that onto TCBS media. In that, we were looking for presumptive yellow colonies. These would be our presumptive Vibrio, and then we would confirm these with PCR. All right, and so once we did that, we had 2 PCR reactions that we carried out to determine the serotype as well as to determine the presence of the toxins, all right? And there are 3 toxins that we were looking for, and all 3 had to be present for us to be able to say that Vibrio cholera was present. And I can safely say, out of about almost 100 samples that we sampled, the Nelson Mandela Bay municipality's water was spotless, no Vibrio cholera. but we were able to detect another Vibrio variant or Vibrio species, Vibrio mechinkovi, which is usually infects immunocompromised patients, causing bacteremia, sepsis, and sometimes even death. And it's being rated as an emerging pathogen. So that is something that the city authorities would have to watch out for. We're also looking at Mycobacterium tuberculosis and the complex, which is made up of Mycobacterium bovis, Mycobacterium africanus, Mycobacterium caneti, as well as Mycobacterium tuberculosis. All these bacteria make up the Mycobacterium tuberculosis, which are responsible for tuberculosis, the disease tuberculosis. And obviously, we know that tuberculosis is a very, very serious condition and disease, particularly in sub-Saharan Africa, where it is exacerbated by the number of HIV infections. It has already been demonstrated that we can actually also survey and we can track the types of species that are prevalent in a particular area, as well as the TB drug resistance in wastewater. And so we are now also doing this at a selected wastewater treatment plant in the Nelson Mandela Bay. And we've already confirmed in our preliminary results the presence of Mycobacterium tuberculosis as well as Mycobacterium bovis, but the other members of this particular complex are present, and therefore we can at this stage postulate that they are not heavily involved in the infections that are present in the city. The last portion that I will mention of the studies that we are doing is antimicrobial resistance. Again, this is being termed the silent pandemic, all right, because it is happening in silence, and I think the last speaker touched a little bit on that as well. And so it is a global concern, and resistance to drugs may be naturally intrinsic. In other words, there are natural mechanisms that that these bacteria have to resist drugs, or they can acquire these from other bacteria in the environment. In wastewater treatment plants, usually resistance will develop because bacteria are constantly being exposed to sub-inhibitory concentrations of antimicrobial drugs. I think there was a speaker yesterday who spoke about the presence of tetracycline in the water, and these are present in sub-inhibitory concentrations, which eventually lead to drug resistance in these organisms. At the moment, we are screening 2 wastewater treatment plants in Nelson Mandela Bay for the presence of drug-resistant bacteria and antimicrobial resistance genes. I'll just again highlight one of the organisms that we've managed to isolate. This is Staphylococcus hemolyticus, and this strain was determined to be resistant to at least 8 antimicrobial drugs. All right, so it is a multi-drug resistant specimen, species, resistant to your penicillins, your ciprofloxacin, your clindamycin, erythromycin, all the way to the tetracycline that we heard about yesterday as well. So again, this is a cause of concern, particularly in individuals that may be immunocompromised. And again, if we are running out of drugs to treat these organisms, this can definitely lead to the next big pandemic. So in conclusion, we actually can see that wastewater surveillance provides a tool for monitoring different pathogens in wastewater, and this obviously informs and can help to warn and inform local government about impending health outbreaks. And because wastewater treatment plants service specific communities, information from this can be used to trace, may be used to trace and isolate individuals that are actually infected, making it easier to get them the help that they actually need. So if there's one thing that the pandemic has taught us, is to be proactive, and that is exactly what we are doing. I'd like to thank all the different individuals that have contributed to this work, and these different organizations for funding the studies. Thank you.
All right. Thank you so much. Is there anyone who have a burning question for him? Oh, okay.
Thank you so much, Brava, for a nice presentation. I have 2 questions for you. The first one is, Given the type of samples that you are working with in your labs, can you tell me what are the measures that you take to ensure that you do not get contamination from the samples that you are working with? That's the first one. The second one is, I heard you mentioned tuberculosis and the resistance of the antimicrobial resistance, as you termed it. Are there any factors that leads to XDR and MDR tuberculosis other than someone not attaining to their treatment? Are there any other factors which contribute to the development of extreme drug resistance, TB, and multi-drug resistance CB? Thank you so much.
All right, thank you. If I got your first question correct, is what measures do we take to prevent ourselves from getting infected? Is that right?
Yes.
Okay, so we work with PPE. All right,
Yes.
so protective clothing. We treat everything as potentially pathogenic. We work in a level 2 laminar flow hood with all our samples to make sure that we do not get contaminated, and we have disposable aprons and suits that we also work with to make sure that we don't take it home with us or ourselves getting contaminated. And then all of us, we're obviously vaccinated before we started working on the project as well.
All right, thank you.
All right. And then the next question.
Yeah, the next question was, I'm not sure how far you went with your tuberculosis studies. Tuberculosis is very prevalent in our community. So I'm interested in knowing if the development of XDR, the multi-drug resistant and that type of TB, is it tied to only someone not attaining to their treatment strictly, or there are any other factors which contribute to that?
I think that probably is the major factor. There could be others, perhaps some type of transfer that is taking place, some horizontal transfer where your resistance is being conferred from one organism to the next organism. That could also be another way, which is why this type of work is important, because in your wastewater treatment plants, that horizontal transfer can actually take place where resistance plasmids can be transferred from one organism to the next. So that might be a reason as well.
All right, thank you so much. Let's give him a round of applause. Our next speaker for today is P. Chigoli. Did I pronounce it right? Okay.
Good afternoon, everyone. My name is Portia Chigodi, and I'll be presenting to you a topic which says mapping crop in a Levubu area using Sentinel-2 and Landsat 8 data. So throughout the conferences, I've heard speakers talking about the food security. So it's a serious global challenge that we're experiencing, and it's one of the challenges that one of the SDG Goal 2 is trying to address, which aims to end hunger and also achieve food security. So like I indicated, it's a serious problem that we're experiencing. So there is a heavy reliance on food production. So we all eat and we all depend on the food, on crop production as the main source of food. So the UN projected a population growth of 9 billion by 2050. So you can tell by such a projection that we'll have a serious challenge in terms of heavy reliance on food production. So the projection that was projected, it will be probably around 83 million population growth per year. So we need proper management of crop production and also proper monitoring of such crop production so that we are able to maintain food production. So one of the issues that we can use in terms of monitoring the food production is by crop mapping. So crop mapping will assist us in developing strategies that can assist us in food security, and also it provides crucial information in terms of guiding government and also agricultural producers in terms of the techniques they can develop and also how they can better manage the crop production. So previously, there was a heavy reliance on traditional cropping method, so crop monitoring method. So one of the traditional method was sensors and also through communicating with the farmers. So that kind of method is effective, but it's time consuming, and it also it doesn't have the spatial coverage. So with the recent advancement of remote sensing data, it's something that we can use, and it has got spatial coverage, and also it offers advantage that you are able to monitor areas where you are not able to monitor, you are not able to reach compared to the traditional method. So it offers such advantage, and also that it offers advantage of you are able to monitor your area frequently, depending on how frequent you want to monitor the area. So it offers spatial resolution and also temporal resolution. So the aim was to map crop types in Limpopo area using Sentinel-2 and Landsat-8, and one of the objectives was to determine and characterize the spatial distribution of crops, and also to assess the potential of Landsat-8 and remote and Sentinel-2 in understanding the spatial distribution of the different crops in Limpopo, and also to determine the potential of of random forest algorithm in classifying different crops using Sentinel-2 and Landsat-8. So the study location was in Limpopo, which is found in Limpopo, and in Limpopo, it is dominated by subtropical crops, and only few citrus crops. So the data set that we used, I used remote sensing data, I used Landsat-8 and also Sentinel-2, and also I used the ground data. So the ground data was collected around November, and the points that were collected were 425 of 8 different land use classes, which composed of crop types and non-crop types. And also the Landsat-8 and Sentinel-2 images were collected during summer. It was summer images that were taken and were used. So the image were processed, were atmospherically corrected. So with the Sentinel-2, the image was corrected using Snap. We're using Snap, using a plugin called Centico2, and with the Landsat-8, the image was processed using NV, using Flash algorithm. So the image were classified using random forest supervised classification, which is also a non-parametric method, because with the non-parametric, the distribution of your sample is not assumed. And it was trained using the R programming language, and with the algorithm. So one of the advantages of remote sensing is that it provides you as a user the advantage of defining your number of trees and also the variables in splitting the modes. So the 70% of the data was used for training, and 30% was used for testing. So accuracy assessment was performed for the study, and then test sample was used, it was done by using test sample to using confusion matrix and kappa coefficient. And so with the kappa coefficient, with the confusion matrix, sorry, so you are able to identify the errors that has taken place in when you are classifying the image, or the misclassification that might have occurred when you were processing the image. So the results of the study shows that both Sentinel-2 and Landsat-8 achieved overall accuracy of more than 70%. And they were all able to distribute between different crop classes. So in terms of band's importance, so the level of the band's importance measures the mean decrease in accuracy. So the higher the mean decrease will also mean that it will have the band would have contributed more. So in this case, in both images, the bands that contributed were different. So with Sentinel-2, the bands that contributed the most were red, green, blue, and near infrared. So on Landsat-8, the bands that contributed the most were coastal red and green. So the least contributed bands on Sentinel-2 were the two SUI bands and also the reg ends band. So what it means is that if you were to remove the bands that didn't contribute much from the model, it wouldn't have the impact on the results of your study. So the overall accuracy of both Sentinel-2 and Landsat-8 were 77% and 87%, and the kappa efficiency was 71% and 84% for both Landsat-8 and Sentinel-2 respectively. So with both, on both the images on both Landsat-8 and Sentinel-2, the crop that that had the highest user and producer accuracy. Okay, Macadamia Nut had the highest user and producer accuracy on both Landsat-8 and Sentinel-2. And maize had the lowest producer accuracy, and also mango had the lowest user accuracy on Landsat-8. So on Sentinel-2, it was only maize that had the lowest user and producer accuracy. So the study showed the potential of using Landsat-2 and Sentinel-2 and Landsat-8 image in mapping crops in Livobo area, although the area is characterized by, it's a heterogeneous area. So they were able to produce the accurate result, and Sentinel-2 results were better compared to Landsat-8 results. And the random forest was able to produce accurate result, is also observed on the literature review. When I was doing the literature review, some of the studies that I reviewed showed that random forest had the potential in mapping crops or different crop types. So what was the most important observation in the study was that in some of the literature review, in some of the literature that I reviewed on the paper, they were saying that the red-edged bands performed better, but in the study, red-edged bands didn't perform well as compared to the other bands. Okay, so the objective of the study were achieved, and the study showed the potential of remote sensing in monitoring, crop, and also in deriving and distinguishing different crop types. So it also showed the potential of random forest as a machine learning classifier in terms of deriving accurate results. And the results will also assist in planning. Like I said when I started that we've got a serious challenge of food security. So the results will also help decision makers and the funders and also people who sponsors, because sometimes when people are sponsored, let's say you're sponsored with money to go and plant a certain type of crop, the sponsors might not be aware of the of the crop that you planted, but with this kind of results, we are able to say if the crop that you were meant to plant, you planted the right crop. And also it contributes in addressing the SDG goal too, like I said in the beginning. These are some of the references that I cited in this study. And I would like to thank the organizers of the conference and everyone who sponsored the study. And thank you.
All right, thank you so much. Is there anyone have a question for her? You again. That's nice.
Thank you so much. Before I ask my question, can you confirm Lancet as well as Sentinel are machine algorithms that are already existing, right?
They've got what?
I'm asking if Lancet, the Lancet and Sentinel are the already existing machine learning algorithms.
No, Lancet and Sentinel are remote sensing data. So it's satellite imagery. It's not an algorithm.
Okay, so what algorithm, what machine learning algorithm did you use?
I used random forest.
Random forest, oh, okay. So my question is, do you think this system that you just presented now can be used to monitor real-time deforestation?
Okay, so with real-time deforestation, the challenge is you only gonna get Landsat image has got a temporal resolution of 16 days. So you only get a Landsat image over the same area after 16 days. So you can do that, not real time though, but near real time. You can do such monitoring when you use other satellite imagery. If you combine it with other high resolution imagery or other satellite imagery, you are able to monitor real time, not real time, near real time, sorry.
Okay, all right, thank you.
All right, is there anyone who have a question for her? Okay.
Thank you for a very informative talk. My question is also on your Landsat 8 and Sentinel-2. You said they both achieved over 70% accuracy. But then one of your statements, you indicated that Sentinel-2 was better than Landsat 8. Can you elaborate on that, and how did you come to your conclusion that that one was better than the other one? Thanks.
Okay, maybe if you notice on my slide, I had the total percentage of each, the overall accuracies of each sensor. So with Landsat, I think it was around 80, I think it was 82, 83, and then Sentinel was around 87% overall accuracy, if I'm not mistaken.
All right, thank you so much, everyone. Let's give a round of applause. All right, our next speaker for today is Anne Ngobeni.
Okay, good day, everyone. My name is Ntabiseng Majomani Nangobeni, and I'm going to be talking to you about the in vitro effects of environmentally relevant concentrations of parananophenol and selected pyrethroids on a mouse Sertoli cell line. So I'm not sure if you all are aware, but male infertility is on the rise. And this is because of the exposure to toxic environmental chemicals that may negatively affect male fertility. And also the increase in use of these chemicals for industrial, climate, and agricultural activities may contribute to the increased exposure. We were not exposed to these chemicals a long time ago, but now that there's advancement in chemical industry, we are seeing increased exposure to these chemicals. And they are called EDCs, endocrine disrupting chemicals, because of their potential to disrupt the endocrinal system or the hormonal system function in both humans and in animals. So endocrine disruptor are defined as exogenous substances or mixtures that alters with the function of the endocrine system. So, like I said, EDCs may lead to increased risk of various health issues, including reproductive disorders, which is the one that we're going to talk about today. And then also it can affect cognitive impairment, as well as neurodevelopmental issues, especially in fetus that are exposed to the cells through their mothers, when their mothers are still pregnant. And then also it may lead to obesity and metabolic disorders, as well as cancers that are related to hormone dependent. So EDCs may have estrogenic and anti-estrogenic as well as anti-androgenic properties, and may play a major role in male reproductive dysfunction. These are some of the sources of exposure to EDCs. We are exposed to the EDCs from agricultural farming products, consumer products. We are exposed to these things through medicine and healthcare products, as well as pollutants. So some of the ways we are exposed to that you can relate to: diet through meat, the meat that we eat, dairy products, fish, pharmaceutical compounds. We are exposed to this through drinking water, and as well as aquatic sports like swimming. And then we are also exposed to this. We can be exposed to this through the air that we breathe if it's polluted, and as well as dermal contact through the creams that we use, and cosmetics, as well as pharmaceutical products. So in males, normal and efficient spermatogenesis or production of sperm is dependent on a normal functioning of Sertoli cells. Now, these are the cells that we expose the chemicals that we studied to. So Sertoli cells in the mammal tissues of males are the targets for FSH because they have that specific receptor for this FSH, which is responsible for production of sperm. If there's any impairment of Sertoli cells, then there'll be dysfunction in sperm production, as well as reduced semen quality or sperm quality. So some of the common causes of infertility are non or occupational exposure to this harmful environmental contaminants. And then there's been a lot of studies that have linked the decline in semen quality or in sperm quality to exposure to pesticides. The pesticides that we use, which are, some of them are pyrethroids, which is the one that we studied, as well as nanophenol. So pyrethroids are mainly used in malaria control programs or agricultural and urban areas. And then nanophenol is the chemical or the primary, so its primary role, use is in manufacturing of plastic or rubber. So that's the compound that makes plastic flexible. And then also it's a surfactant in pesticides formulation, or a carrier molecule in pesticides formulation. And then we use pesticides widely in agricultural sectors, obviously, to enhance production of food, of crops, and also to preserve, store, and transport your fruit so that it reaches the shops still fresh. And then we also use this chemicals in our houses to prevent diseases transmitted through vectors. We use this chemical to like kill mosquitoes and flies. And some of these some of these products have these chemicals. So it is important also to mention that it does not only interfere with the hormonal system, but it can also induce oxidative stress. And then, so that's by increasing the production of harmful molecule in the cells. So that molecule is called reactive oxygen species. And when this molecule is, when they're elevated, they can lead to cell damage. And then in Sertoli cells, exposure to EDCs such as these two chemicals, paranonophenol and pyrethroids, can cause the imbalance of the ROS and then cell defense, and then that can lead to oxidative damage to the cell components, resulting in obviously impaired spermatogenesis and then ultimately male reproductive issues. So the aim of the study was to investigate the effects that these two chemicals would have on the mouse Sertoli cells, and we exposed the cells to concentrations that are found in the environment in South Africa. So we did that by assessing the toxicity or cell viability after exposure to these chemicals using MTT-assay. And then we also assessed the level of ROS, of reactive oxygen species, and then we also looked at the morphological changes of the cells that are exposed to or were exposed to these chemicals using the H&E staining technique. So this was the method. Briefly, we see that the cells in a 96 well played. Let's see if we can find it. Okay, it's not working. So we exposed the cells. We seeded the cells and then we incubated for 24 hours in 5% CO2 at 37 degrees Celsius. And then after a day, after 24 hours, we treated the cells with the compound and then incubated again day 2. Day 3, we added MTT, another further incubation for 4 hours, and then we read the reaction using a spectrophotometer. And then the second part was measuring oxidative stress. We got a kit for that. Then we seeded the cells, stained, and then we exposed to these compounds, and then we analyzed using a fluorometer. And then the third part was to look at the morphological changes that the cells had after exposure to these chemicals. And then the results. So toxicity wasn't affected that much, except with the concentration that we exposed to, except at 100 milligrams per liter. That caused total cell damage. But the good thing is that we haven't found levels as high as 100, so we are still safe there. And then with the ROS, there was minimal ROS production at the concentrations that we exposed to the cells to. And then with the morphological changes or morphological damage or cell damage, there was an increase in percentage of cells that were demonstrating abnormal morphology, cells that were exposed to this chemical did show cell damage, and as well as these chemicals being cypermethrin, deltamethrin, and reg transpermethrin. Sorry, I'm just gonna put my jacket here. Oh, okay. So I was still saying, so there was definitely cell damage after exposure. And this is what the cells looked like. So as you can see, this is normal. This is a normal control. The cells are nice and round, and they are in the resting phase, which is called interphase. And then there's a couple of cells dividing over there, but this is normal. So after exposure to these chemicals, you can see this does not look good. There was, the cells are in distress, plasma membrane blabbing, and then as well as chromatin hypercondensation, which indicates that the cells are in distress and they are dying. And then there was this one, cell elongation as well, which speaks to the disruption of cytoskeleton. So the summary, after exposure to these environmental concentration of the selected compound, we saw 80% of the cells remaining viable, but I mean, you want them to be at 100%, so that is worrisome. But there was low production of ROS, indicating minimal cell damage, which is good, but and again, you want your cells to be 100%. And then, obviously, as we see from the previous slides, that there were aberrant morphological alterations. And then it was also important to note that exposure to higher concentrations of para-nanophenol resulted in reduced cell viability. So, in conclusion, the study demonstrated that exposure to these two cells may lead to detrimental effects to mouse Sertoli cells. And then, although the compounds did not induce ROS, morphological alteration were observed. So we recommend that the future studies are conducted in order to explore the effect of prolonged in vitro exposure to the environmental concentrations that are found in South Africa. And the reason we didn't see a lot of damage might be because these are cells, so we can't expose them to the chemicals for a longer period. With people, we are exposed to these chemicals for a long period, but we couldn't expose the chemicals to the cells to these chemicals for extended periods because the cells would die. So, but we are exposed for long periods. So there should be. We recommend that there are studies that would explore the effects of prolonged exposure, in vitro exposure to these chemicals. And then also future studies are necessary to determine whether the chemicals will have same effects on human Sertoli cells. I mean, we used the rodent models, which do provide great insight into the potential of reproductive effects of these two chemicals, but it will help to use human Sertoli cells in order to confirm the relevance of this finding for human reproductive health. And I would like to thank the project funders, NRF and Tshwane University of Technology, and then as well as the conference organizing committee. Thank you.
All right, thank you so much. I'm not too sure if there's anyone who have a question for her.
Thank you so much. I have a condition on the endocrine system. I have a thyroid disease. So I'm interested in knowing what is the effect of these chemicals on the thyroid, specifically on the thyroid gland. That's one. Secondly, I wanted to know if the Sertoli cells in the chest are regenerative. I learned that organs like livers are regenerative if damage has occurred after some time. Depending on the amount of damage, they are able to regenerate back. So, do we have something like that in the Sertoli cells, or once they are damaged permanently? Thank you so much.
I speak under correction. I'm not sure, but Sertoli cells are you can, I mean, they are regenerative, but you must remember spermatogenesis takes place every day. And then the sperm that you see today is sperm that was produced maybe 3 months ago, and it undergoes that cycle of maturation.
Okay.
So at that time, you want the cells to be healthy so that they can nurture, because the primary role of these Sertoli cells is to nurture the sperm cells.
Yes.
So if they are not okay at that time, if they are damaged, then the sperm will not be, it's not going to be nurtured, and then the sperm will not mature.
Okay,
Yeah.
all right, thank you.
Thank you.
All right. Is there any question for her? If there is none, can we just give her a round of applause? All right, our speaker for today, the next one is S. Sibanda. I think they are asking that if we can just stand somewhere here, because now you're affecting the... Okay, so you're just affecting the... Yeah. So if you can just stand here, make sure that you don't go over the whole, what do you call this? Yeah.
A pleasant afternoon to you all. I am Sipos Banda, and I'm presenting my topic entitled Impact of Phytochemical-Based Hydrocolloid Dressing on Wound Healing. This is a comparative review. All right, I'll take you through the introduction up till the conclusion. Let me start by posing a question to you all. How many of you guys have ever had wounds? Obviously, almost like everyone, right? Every one of us have been burnt at one point in time. That's true. The next thing, what type of medicine did you use? Most probably complementary medicine, the standard medicine that we have in the system. Right. So for the standard drug that we have, we've got one that is known as silver sulfadiazine. And here, in this study, we're looking at plant-based, phytochemical-based medicine, right? Phytochemical-based hydrocolloids. Ethnobotany is one such way we can find these plant materials. You'll find that in the system already, we've got up to 40% of these phytochemicals being used in our current modern synthetic medicine, sometimes as mimics. Why these, looking at these plant-based materials? They are readily available, and even for their formulation, they're biocompatible, biodegradable, and even sustainable. You look at the healing process. The healing process of wounds is mostly complex and aggravated by different factors. But however, when we're looking at the use of plant materials, it helps by speeding up the healing process. And also, the advantages of using plant-based bioactive compounds is that you'll have triple and sometimes quad effect, we are looking at the antimicrobial activity, the antioxidant activity, the anti-inflammatory activity, and also, at times, the tissue regenerative activity. So that means it will assist with cell proliferation. So researchers are looking into embedding these plant bioactive compounds and formulating them into hydrocolloid bandages. And these are used for mild exuding wounds. So for our problem statement, we are saying wounds, if left untreated or if they are being hindered by other factors, they can progress and become chronic. And once they become chronic, the right treatment measurement is hard to find. You'll find that it is a global burden. There's a global burden of wounds, which is estimated from up to 40 to 50 million surgical wounds, 8 to 10 million leg ulcers, 7 to 8 million pressure ulcers, and 7 to 10 million burns worldwide. And this is was reported for the year 2009. And to date, we are saying the chronic wounds are reported to affect up to 40 million patients globally. And then coming down home to the sub-Saharan Africa, we've got over 1 million burn or wound injuries, which are recorded annually. I'm sure we are aware of the Joburg fires that occurred last year. All these are effects that result in wounds. For justification, what has been done? You'll find that there are a number of dressings that have been made, a number of dressings that are already in the system, some that are synthetic. We've got creams, we've got hydrogels. We also have anti-inflammatory drugs that are used to support the healing process. However, the downside is that with these drugs, most of them, they are expensive. Apart from being expensive, some of them have side effects, and these side effects include cardiovascular failure, damage to the kidney, skin thinning, the list goes on. The major challenge is that for different types of wounds, there is need for different healing conditions. So, in essence, as a result, mostly there is different types of dressings for the optimal wound healing to occur. So what is the proposed solution? Is the use of phytoconstituents, phytochemicals. Why? Because these are safe and these have got multiple biological activity. We are saying for a particular wound, maybe this is a pressure ulcer, that is being caused by systematic inflammatory activity, right? If you put a hydrocolloid that has these triple effects, that anti-inflammatory parts of that hydrocolloid, plant-based hydrocolloid, will come into action and help the wound to heal. So this type of wound dressings is like one type fits all. So the aim of this study was to conduct an exhaustive search of existing literature on plant-based hydrocolloid dressings for wound healing to evaluate the dressing efficacy in comparison to standard dressings. So for our methodology, we used a systematic search criteria whereby we made use of keywords. I'll show you the keywords in the next slide. And this was to identify clinical and randomized control trials from the year 2019 to 2024. The data appraisal was done by analysis, whereby the authors reviewed the data quality. For the PICOM framework, we are saying our population were wounds, all types of wounds. Our intervention, we were particularly selecting articles that looked at phytochemical formulated hydrocolloid dressings. And for our comparison, we looked at those that are non-phytochemically based, and the outcome of choice was wound healing based on wound contraction and histological evaluations. So these are the keywords and the databases that we used, our search terms that we used, and the synonyms. So in general, we looked at wounds, dressings or bandage, plant extracts, phytochemicals, and so forth. And the databases that we looked at were Web of Science, Scopus, PubMed, Science Direct, Cochrane, and Embase. For our selection criteria, we included only original research about randomized trials and clinical trials which investigate phytochemical formulated hydrocolloid dressings. And these were for specifically all types of what? Of the wounds. And for our exclusion criteria, we looked at intervention produced by non-plant-based hydrocolloid dressings or studies that compared intervention other than polymeric dressings. And then studies that, or formulation characterized of plant-based hydrocolloids lacking the wound healing assessment were excluded from the records, right? So for our data extraction, we made use of the PRISMA guidelines. And then to assess the literature, we specifically looked at the wound type, the test method, the test subject, and these were used to compare the effectiveness of the particular hydrocolloids. And then in addition, the wound contraction area and the histological evaluations were looked at. So for our key results, out of the search that we carried out, we found a total of 866 articles. And after excluding the articles due to alternative dressing types and a number of exclusion criteria, we went on down to only 4 articles that looked at hydrocolloid dressings. So from the 4 articles that we got hold of, we looked at the test method, the type of the wound, the subject, the polymers, the polymer base, the plant bioactive compounds, the characterization methods that they used to formulate, and also the control. So you find that in the different articles, we had different types of systems that they used, especially for the characterization and the polymer bases. And these had an effect on the results that we obtained. So for the findings, the characteristics of test subject and wound type as a factor of wound healing. You'll find that the age of the rates that were used affected. We've got 4 of the 4 articles, 3 of the articles made use of rates that were 8 weeks old, and then one of the articles made use of rates that were 12 weeks old. And then you'll find that the age difference has an effect on the healing process. Also the weight and the type of the rates that were used. The good part about all these 4 articles is that the type of the rates that were used were similar, and these were the adult male Sprague-Dawley rates. Also, the type of the wound is something that we also looked at. So they used pressure ulcers and acute wounds. And with these 2 types of wounds, the rate of healing also differs. We also had to look at the plant hydrocolloid formulation and its mechanism. So common compounds such as catechin, rutin, quercetin, and all the other flavonoids that are there, known to have wound healing activity on their own. And these were common within the plant materials that were used. So that's indicative of the fact that as research, we have to focus more on the flavonoids and alkaloids, as they may be our go-to for wound healing. So accelerated wound contraction, epithelialization, increase in tensile strength was noted, and collagen formation and also vascularization was noted. The design and characterization of the hydrocolloid was also something that was sensitive in this study. So you'll find that most of them looked at the morphological characteristics of the hydrocolloids that they formulated, and most of them made use of the scanning electron microscope. And for the chemical analysis, there were studies to do with the FTIR. And for physical analysis, they looked at the MVTR, the tensile strength, and the drug release. And others also went on to look at other methods of characterization for physical analysis. So this was done to ensure that the potential of the dressing in conferring wound healing activity was up to par. The biological mechanism of action of the formulated phytochemical base hydrocolloids was also looked at, and these were the wound contraction and the histological evaluations, as I mentioned. This picture just shows the histological evaluations that were taken in one of the papers. So the impact of phytochemical-based hydrocolloids, you'll find that the flavonoids and the alkaloids reported to have expedited the wound healing. And the articles reported an enhanced mechanism of action. You'll find in the picture to our right, there's a rat that had been inflicted with burns, and by day 19, the burn had healed due to the action of the plant-based materials. So as an aspect of interdisciplinary research, these studies look into coming up with work from traditional healers, diviner diagnosticians, that point to them the type of plants that needs to be used, and then that goes on to analysis, phytochemical extraction, phytochemical analysis, and the formulation. And with this study, we are saying the preclinical studies must be followed up by clinical trials in order to assess the action of the plant hydrocolloids on humans. And policymakers and funders should also be made aware of the current work in this field in order to prioritize this research. Also, the good part is that it is accessible, affordable, and sustainable, and it has economic and social impact as it aligns with the SDG 3 and the SDG 9. Right. In conclusion, the study showed the phytochemical-based hydrocolloid research is still low, as we only found 4 animal-based studies and 1 clinical trial. And the success of phytochemical-based hydrocolloid is due to its characteristics, which are basically the anti-inflammatory, antioxidant activity, cell proliferation activity, and antimicrobial activity. Thank you.
All right, thank you so much. Is there anyone who have a question? Okay.
Thank you so much for the wonderful presentation. So I would like to, I'm interested in one of the things you found in the study regarding the weight versus rate of healing. What was the main finding in terms of the recovery rate of different weight?
Okay, thank you for that question. So fortunately enough, for this study, the rates were ranging in the same weight brackets. As I said before, 3 of the studies had weights that were, had ages that were similar. And for rates, mostly the ages range also go along with weight. So with those studies, the results were just similar. The difference now was then to look at the particular compounds that were used. Because also we have to look at the compounds that were used and the base material, the base polymer that was used. So because of the weight, it was not really an issue.
All right, thank you so much. Can you give me a round of applause for her?
Thank you.
All right, our last speaker for today is M. Ngayana.
Good day, colleagues. Good day, colleagues. My name is Mbulela Ngayana. I'm a PhD student with the University of Mpumalanga. I'm here to present part of my current ongoing study that I'm doing for PhD that looks at the optimization of nutrient management for pigeon pea in South Africa. So we are just trying to develop some nutrient management strategies for the crop, and also to look around how the crop does. So this is the outline of my studies, just a short introduction, and also how the pigeon pea does in terms of the food systems, and also we look at how is it relevant into sustainable development. And then I will share just briefly what the study is about, because it's part of the trial that we are still trying to get it concluded. So the pigeon pea, I'm not sure if many of you have seen the crop. It looks like those nice, beautiful flowers that I can't share with the ladies, because otherwise I will be pushing the country into food insecurity. So that's how the crop looks like. So in terms of the importance of the crop, I think we have seen and how the world has been affected, or it's suffering in terms of the food insecurity. So we are just trying to see, so the crop, it shows that it's viable in terms of how it can go to resolve the issues around food security. You'll notice that most of the times, in terms of, because it's a legume crop, so most of the legumes have been bred throughout the years to be more reliant on the nitrogen and also fertilizer applications. So this crop, it shows a potential that it can, well, most of the legumes are able to fix nitrogen, but this one, it shows that it can do that, but it also uses phosphorus efficiently, even on degraded soils. So this is what we will share at the moment, and then we look at how it contributes into food security. I think we've all seen some of these protein sources. As much as one of them, because of my pot belly, I can't claim to be using it. We have seen how even from last year, how the avian flu, in terms of the chicken, it has affected the prices of eggs and the meat. And also how, in terms of when you're looking at the beef prices, how they have increased. Also looking at the issues of climate change, because it is said that animals generally contribute around 14 to 16% of the global, I think of the global warming. That's how much the animals normally contribute towards that. So now this is why we are trying to preach the use of legumes as a source of proteins. Also, you can even see in terms of the price range for dried beans and also legumes, it's only around 50 cents per gram of protein that you can spend on your legumes as compared to your beef, which is around more than 3 rand. So pigeon pea, it plays a huge role in terms of the food systems. It can be used in rotation with other crops. It can fix nitrogen. As I've said, it also can use phosphorus in low phosphorus soils. So it's a versatile crop. It also can be stress tolerant in terms of it can perform better under stress soils and also under dry conditions. And also, I think the crop, it contributes towards SDG 2, which is to contribute towards zero hunger, and also SDG 13, where it's working towards in sustainable climate action, which then through the use of the crop under various climate smart agriculture practices. So it also improves soil health and also reduce the chemical fertilizer use. So in terms of the problem that we have observed is that there's generally a lack of commercial cultivars. So as part of the study, we are trying to because there are no registered cultivars for most of these crops that are normally identified as, I'm not sure if they prefer identifying them as poor man's crop or forgotten crops or something like that. But due to lack of registered cultivars, these crops, they generally are difficult to find seeds of these crops. So you find that most smallholder farmers, they end up unable to produce these crops because it's very difficult to find. So we are trying in this study we are using, you'll see we are using two cultivars that we are trying to get them registered for release. And also in terms of the nutrient management. So these crops, it's difficult to actually go and recommend to say, plant this crop because there are no general guidelines on how you apply your nutrients and how much nutrients can the crop actually use. So in terms of the study, we are just trying to explore how much the different nitrogen and phosphorus levels that can be used under different climatic zones. As part of the primary objectives, we are looking at nitrogen and phosphorus, and also in terms of the cultivars, we are also trying to identify how the crop can be used in a sustainable manner. So this is the current study, but the overall project, we are doing it under 2 climatic conditions. We are looking at the high veld region and also at the low veld region. So these current results here are only from University of Mpumalanga, which is a low region. It's characterized by very low beans or beans production areas. So we are looking at different levels of nitrogen from 0 to 56. I think from the literature or the previous studies, the normally recommended rate is around 28, but as much as we are taking it from other studies, there are no general recommended rates for the crop. So we are still trying to establish, even for phosphorus, we are trying to identify which levels are more suitable for the crop. We are looking at 2 cultivars, and then we will measure different growth parameters. As you can see, my babies were still very young and innocent there, but now they are problematic. They are old and problematic. So as part of the small preliminary results that I will share with you, so we have observed for the 2 cultivars that we are working on, we generally have seen that the first cultivar, it generally grows upright, so it had higher height in terms of height growth, but the second cultivar, which it grows mostly in branches. So you can see even on the leaf area, the cultivar CV2, it has a higher leaf area that it covers because it grows with more branches to the side, similar with the number of branches as compared to the first cultivar. And then in terms of the yield response, we have witnessed that in terms of normally your yield will be higher at N28, but if you go above that application rate, there is no significant that exists in terms of the yield that you'll get. But in terms of the use of the actual nitrogen that you have applied, we have witnessed that the crop actually uses very low nitrogen from what you have applied in the soil. So it's similar with the phosphorus because we applied different rates, but there was no significant difference in terms of the actual use of the phosphorus that we applied. So I think I need to indicate that we are still going to measure how much is then fixed in terms of the phosphorus, and also in terms of we are just send the samples for analysis for these results to quantify how much as you can see, there is some nodulation that happens. So we are trying to identify the different types of species that the crop can use in terms of the nitrogen fixation. We are trying to quantify how many species exist within the soils, and also how much nitrogen is actually fixed by the crop. So we are still working around those results. So I hope that next time I come and share, I will share the full picture of what happens actually in terms of the nitrogen and phosphorus behavior of the crop. So we wanted to, on this study, we want to recommend based on 4 things. So for smallholder farmers, it's the adoption of the crop as a low cost. If we manage to get the proper seeds, and also at a lower rate prices if we manage to release those cultivars. And also in terms of the nutrient management, we are trying to promote the efficient use of nutrients by the farmers. And also in terms of the policy recommendations, we are trying to get into a place where we can actually have proper guidelines for production of this crop, because as part of the study, we are also going to look at a greenhouse study where we are actually looking at in terms of is the crop able to, because it's been used in agroforestry in some areas. We are going to do a comparison study in terms of how much carbon sequestration can the crop do. So yeah, in conclusion, we just want to provide some insights in terms of the nutrient management of the crop, and we are hoping that the full results when we conclude the study will give us a better picture. Thank you so much to everyone.
All right, thank you so much. I only have 2 minutes for him. Is there anyone who have a burning question? Okay, please ask the question, because you do have.
Okay, I wanted to ask if it's the crop you're talking about, did you find it in specific area in South Africa, or is it a crop that is found in all the provinces in South Africa? Also, is it found also outside South Africa, or maybe in certain parts of Africa?
Okay, now thank you so much. The crop is actually, it is used, but mostly it's smallholder farmers. So in regions like I think it's found in around Zanin, there are farmers who are growing the crop. We also have few in KZN around Tuba who have been uh, growing and using the crop. I think also in Mpumalanga there are few farmers that are doing the crop. So it's here, it's there in South Africa, but it's due to the limitation. It's the availability of seeds actually for the crop to be widely used in that way. But it's also mainly used outside of Africa in places like your Malawi, Tanzania. It's very dominant, and also in places like India, because I think it generally originates from that side.
All right, thank you so much. I would like all the presenters to come up front, then we take a picture, and then we just give them the round of applause for today. Thank you so much for coming and also be the part of this session. It was more fruitful, and I would love you guys to collaborate. You guys are doing a massive work. I'm telling you, you are being blown each and every presentation. Like, my goodness, it's as if I never been in school, because I'm learning every day. Each and every presentation, it was on well prepared, and how you guys presented, I like it. So please, guys, connect on a LinkedIn with each other. I want to see you next time being moderating this session like this. Please, I encourage each and everyone to participate and do more, and also in terms of like collaborating with each other. Thank you so much for the fruitful presentation. You missed out the group picture, unfortunately. Thank you. Bye.
Again, I close my eyes. I close my eyes. Play back, repeating the same old night. Waking up stupid, thinking, what's the plan? I know for sure where it's gonna end. Where it's gonna end. It's a new life coming. I don't wanna share the same things. I don't wanna see me. Or feel the new. Yeah. It's a new life coming. I don't wanna share the same things. I don't wanna see me. Way slowly opening. Give that everybody wants some. Sorry, I'm say you're wrong. Oh, 'cause it's always green on the other side. Always green on the other side. It's always green on the other side. Always green on the other side. Love of the unknown. You're gonna get it and look somewhere else. And you come back alone, you come back, ready to live steady now, but no one wanna talk. Depending on us all, 'cause your mission didn't work out. You didn't know time would slip this fast. Now it's too late. Now it's too late. Now it's too It's always green on the other side. Always green on the other side. 'Cause it's always green on the other side. Always green on the other side. Oh, yeah. straightforward, but do you everything? A part of me is in you, part of you is in me. Lost a little bit of tension sometimes doesn't hurt. We're best friend often will break us down and shut. Lost, you open your minds, thinking for what you share. Giving me the high that I won't get anywhere else. Watch the nation. What's the nation? Oh, what's the nation? What's the nation? What's the nation? What's the nation? Oh, what's the nation? 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 Oh, what's the nation? Watch the nation. Can't stop. Tomorrow's still on your mind. Are you letting yourself expand? Let the mask fall off your chair. No need to prove anything. Nightmares on my face. Yesterday, slow flame. Here we are one again. Feel it all, unafraid. Yesterday's, same old game. Same. Can't stop. Can't stop. Can't stop. Can't stop. Can't stop. small, so I don't feel important, so I don't waste it all and be wrong about what I want. I know I'm nothing, but my mind keeps lying. Speck of dust, blink your eyes, and we're gone. So let's live. Let's live. You know what I mean? That's right. That's it. Let's live. Let's live. So beautiful, but I'm blind now. Oh, I know. Let's wake up now and not miss it anymore. Born again, better late than never. Wish me luck on my new trips around the sun. Don't let it get you. 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 Let's live. Let's live. Let's live. Let's live. Put it down, no, I gotta zone out to get by. Want some love, I'll try. But once again, I close my eyes. I close my eyes. Play back, repeating the same old night. Waking up, stupid, thinking, what's the plan? I know for sure it's gonna end. Where it's gonna end. Once again I close my eyes. It's a new life coming. I don't share the same things. I don't see me. Or feel got new. Yeah. It's a new life coming. I don't share the same things. I don't see me. Or feel got new. Yeah. It's a new life coming. I don't share the same thing. I don't see it. I feel that new one. Yeah. Oh, I feel that new one. Yeah. I feel that new one. on the other side. Always greener on the other side. Love the unknown. You're gonna get it and look somewhere else. And you come back alone, you come back, ready to live steady now, but no one wanna talk. Depending on us all, cause your mission didn't work out. You didn't know time would slip this fast. Now it's too late. Now it's too late. Now it's too It's always greener on the other side. Always greener on the other side. It's always greener on the other side. Always greener on the other side.
pronouncing it correctly? Sipalana. Am I pronouncing it? Shipalana. Okay. And then it's Ngwenya. Okay. Shipalana, are you still busy uploading? Okay, I think we can start in the meantime because we only had an hour allocated for this. Session. Okay, good afternoon, everyone. And welcome to this session on advancing science and technology for development. We are very excited and looking forward to the presentations to come. And our first presenter is Ms. B. Nkuna. So we look forward to your presentation, and over to you.
Good afternoon, colleagues. Okay, my name is Basani Nkona, and I'll be presenting to you today the models that we are trying to develop to help the smallholder farmers under the title that says Identification of Maize Leaf Diseases using RGB-based images with convolutional neural network and residual network. Maize is one of the 3 produced crops in the world, and it serves as a staple food which has high carbohydrate, and we consume it as starch. Approximately 15 million people grow maize in the sub-Saharan Africa. However, it's only 30% that is contributing to the whole world. So we are affected by abiotic and biotic factors which cause up to 80% of maize yield loss. Smallholder farmers are the ones that are suffering most in this situation because they lack knowledge and resources to deal with diseases. Maize diseases has been treated before based on crop calendar and the application of pesticides everywhere in the field. And this method is costly and time-consuming. With remote sensing now, we are trying to manage the cost and also manage the time of dealing with these diseases in a simpler way. This brings us to the aim of this study, which we are aiming to detect these different maize diseases in the subfield scale using RGB-based images and deep learning techniques. The objective is to identify different maize leaf diseases to create data set for training and testing CNN model and RASNet 50 model. The second objective, we are aiming to develop an app for detecting maize diseases. The study took place in Guyana, where it's a mostly rural area and small-scale farmers are featured. Most small-scale fields, they are between 0.5 to 15 hectares. Okay, our materials and methods, we had 2 data sets that we have collected. The first one, we collected 300 maize leaves, which we took to the lab to identify diseases. And we managed to get 7 categories, which was healthy, maize streak, phosphorus deficiency, north corn leaf blight, south corn leaf blight, nitrogen deficiency, and yellow streaking. The second data set, we captured 1.5 images with a handheld camera, and from there we pre-process it to clean the noise background by using the Clipping Magic software to clip only the leaf of interest out of everything that was in the image. We then use the results from the lab to label all the images that we captured. And then from there, we fed the information to Anaconda using the Python language and split the data into 80-20 training and testing data sets. The data set was then used to run the RASNet 50 and CNN structures. The ResNet 50 it has 50 layers that keeps on running the same leaf until we get the results, while the CNN has only 6. So we wanted to compare these two models, hence we are using both of them, but to see which one is the best, or maybe to see the best performance of them. After that, we did the classification, and then out of the classification, we then tested our model in detecting the diseases. Our results for ResNet 50, we managed to achieve 78.76% accuracy, which the highest classified category with users and producers accuracy was healthy. For both, we maintained 91.6. And then we also got 78.33 users accuracy for southern corn blight, which was much better. Although, if you can see on my right here, the graph there, you see the misclassification, hence the dark color shows us the one that was well classified. And then for CNN, we maintained 71.17% accuracy assessment. As well here, we got 82.82 users accuracy and 76.6 producers accuracy under health category, which was the highest. And then this brought us to our conclusion, where we developed the model to detect diseases. We used the same information to develop an app with these models, and we used the training data that we obtained from the lab to detect now, and we are hoping to deliver this app to the small-scale farmers in Mopani district and also Vembe district, maybe by next month, so they can use it to detect the diseases. And we hope to solve some problems that they are facing in regard to ending hunger and improve food security by managing the diseases at their early stages while they are still growing, and also cutting costs in the pesticides that they are using in the field. Thank you very much.
Thank you for that lovely presentation. I didn't get a set. We have 8 minutes of your time left. So I thought you could answer a couple of questions. I didn't get a sense at the beginning of your presentation that you were developing an app. But it's really cool that you did so. And I was wondering, at the end of the day, when farmers have this app, will they require access to internet? How do you ensure that farmers in rural areas have the necessary support to use an app like this?
Okay. This app, we are developing it in a way that it has to accommodate the small-scale farmers, meaning we are also looking also in the cost and everything. We are trying to cut that. So the app itself, it doesn't need internet. It will be, we are actually going to give them to their mobile cell phones, wherever that will be in that workshop when we go there. So that's what we're aiming. And we are still trying as well to add some information on maybe the specific pesticides for that specific disease that we got in the same application. So the only thing is to give them the training that you take an image like this, and then whatever that information they need, it will appear. And when they go, let's say, to NPK or wherever where they are buying this pesticide, they will just show them and get whatever that they want.
Okay, that's great. I was very shocked by the data that you shared, that 80% of maize yield is lost due to disease. Is that in general or just for smallholder farmers?
As in general. Here I generalize because it's biotic and abiotic. It can be environmental, weather, and everything.
Because that's a
But
very large loss in
yes,
yield.
it is, it is.
Okay. Okay, we have 6.5 minutes. Are there any questions from any of the audience members? Okay, yes, please go ahead. Can we pass the mic?
Thank you. I just wanted to add on to the question that you asked about the app and how it will be able to be used by the farmers. I just want to understand, will they not need some sort of connectivity? Like, is it a free use app that doesn't require any charges, or is it like, I don't really understand the network and connection part and how the app can work without any like consideration of costs. Like, what does that look like?
Okay, the app, it doesn't need any connectivity when you do this thing. It's just that it doesn't need it. I don't know how to put it. But you just take a picture, and then I don't know if you understand. There's this one that we normally use everywhere, Survey 123. We normally develop most of the apps there using Survey 123. It doesn't need any connectivity. Wherever you go, you open it, and then you click whatever that you need.
Okay. I'm very intrigued by this. So I think perhaps a better question would be, could you break down how the app works, like the processing aspect of it? Like, how is it possible? I'm not sure if this is a silly question, but how is it possible to take a picture and have that much information about a plant species show up?
Okay. If you can see here, ouch.
Can we have the presentation back up?
Here. This one, if you can see, I don't see the pointer. Yeah, I can see. Okay, but it's fine. If you can see this screen here, these are the models, the ones that I was talking about. Here you browse where, like in your gallery, in your phone. This is where you browse, and you can see here it's taking any form of the image the way you saved it. And then you browse it and you put it here. Now you come and click ResNet, because I was testing both of them. When you click ResNet, it shows you here it's a maze track. The way the model was trained, this is the result here. And then if you want to see the CNN, again, you come down here. You click and then you check here. Yes.
Oh, we have a question over there. We have 3.5 minutes.
No, thank you so much for the lovely presentation. So I think for me with the app, it's more like Limpopo, it's a resource-strict society. It's like, I think it's not quite clear as to, because you would have small farmers who don't actually have access to phones. So you see like there's a digital divide. Not everybody have an access to phone. I'm not so sure with farmers who actually do not have even access to a smartphone. How are they going to make use of this? Have you thought about catering for such farmers? I don't know. I would love to understand how are you going to cater for farmers who actually do not have a smartphone. And lastly, because I'm not a maize expert, but I'm just interested in understanding what's the prominent disease, maize disease, in the northern part of South Africa.
Okay.
Since, according to literature, since you were doing this, so I'm just trying to understand.
Okay. All right. Your first question was about the mobile phones with the farmers. Yes. We've been engaging with these farmers, and it's a challenge sometimes because you find that you are doing a research in their farm. Sometimes they want you to share whatever that you got in their farm, but you don't even know how to do so. So normally, even this one, we propose this app with them, and they like it like they want it. Then what's the way forward? Some, they are working with their kids in their farms. They will say, you will communicate with this one, I'll get these things through this one. Although now I only engage with few, which now when we deliver this, I'm hoping we can have a workshop that can accommodate everyone. At this point, I am not sure how we're gonna handle that. So the next question is about the most common diseases or disease,
Yes.
maize disease in the north. I would say little studies have been done in the north, but along KZN, Eastern Cape, that's where most diseases have been found, which is maize streak and grey leaf spot, which I didn't get in the north. Yes.
Okay. Colleagues, please help me to thank our speaker. You did very well with time. I hope all the other presenters can copy this fantastic example you have set. Let's move on then to our next presenter, M. Shabalala. Mr. Shabalala, we look forward to your presentation.
Good afternoon, ladies and gentlemen. As introduced, my name is Mandela Chawalala. I am from Mintech, based in Johannesburg. Today I'll be giving a talk titled Green Approach for the Synthesis of Silver-Doped Zinc Oxide Nanoparticles using beta-cyclodextrin and honey for photodegradation of bromophenol blue. So we are looking at using green chemistry to synthesize nanoparticles for water treatment. Globally, we are facing water scarcity and water pollution, which is often caused by either natural disasters or human activities. So my presentation will be based on the figures that have red arrows. So we are looking at the release of dye waste from textile industries that ends up in our water sources, and we're also looking at organic pollutants that mostly come from pharmaceuticals. So the use of chemicals in advanced technology to sustain life is one of the major causes of micropollutants that we find in our water systems. Micropollutants have become quite difficult to remove from water simply because the current systems that we have not been designed to deal with such persistent toxic carcinogenic micropollutants. So some of the pollutants that we're looking at bromophenol blue, which is the dye, and also used as a pH indicator. We have atrazine, which is a pesticide. We have bisphenol A, which is the plasticizer. Rhodamine B is another dye, and then we have that chlorinated phenol at 2.4 dichlorophenol. So most of these compounds are bioaccumulative, they're persistent, and they're produced in high volumes. They often lead to feminization and reduced life expectancy of aquatic animals. So how do we then want to deal with this in this research paper? We are looking at using an advanced oxidation process, which is photocatalysis, which is based on the use of light-activated material. So when you shine your material, your light, mostly sunlight, into this titanium dioxide or zinc oxide that we're going to use, we have the excitation of electrons from the valence band to the conduction band. Once the two are separated, then we're able to generate reactive oxidizing species which can directly interact with the organic pollutants, or they interact with dissolved oxygen and water to produce your superoxide radicals and hydroxyl radicals. So the aim of this study is to use silver nanoparticles to modify zinc oxide nanoparticles in the presence of beta-cyclodextrin and honey as natural surfactants for the simultaneous photodegradation of dyes and micropollutants in aqueous solutions. So the first objective is to synthesize and characterize our main material, which is the zinc oxide. And then we continue to do that and add beta-cyclodextrin and also add honey as a surfactants to increase the surface area and the particle distribution. And then we carry out photodegradation studies of the dyes, the micropollutants, and we also test if they can degrade a mixture of dyes and micropollutants. So our simple procedure that we used to synthesize this was we dissolved our precursors in water where we added sodium hydroxide to adjust the pH, and then we allowed that to run for a few minutes, and then we aged, dry, and centrifuged. And then from there, we used what we call a photodeposition method to introduce the silver nanoparticles. And then we obtained those materials. We then went on to characterize our materials to check if we got what we want or not. So the first technique that we used was UVVs to check the optical properties of the material. And then we were able to calculate the band gaps, and we found that at 3% loading of silver nanoparticles, that's where we have the lowest band gap for the three materials. So the first one, the zinc oxide, ZN0, is the pristine zinc oxide. And then the zinc oxide with the C is the one we use cyclodextrin, and the H we used honey. So looking at the band gap that we have at 3% loading, it means this material can be activated by direct sunlight instead of using UVVs. We then went on to check our Raman spec, which further confirmed that we successfully loaded our silver nanoparticles, and the material that we produced was zinc oxide. We then checked with XRD to complement what we got from our Raman, and then we found that indeed what we got from Raman is correct. And then we calculated the crystalline size, which we found that the smallest particle size that we could obtain was 9.38 nanometers, and this was when we synthesized in the presence of honey. From there, we went on to check the photoluminescence properties, because what we're looking for is that once you generate those electrons and holes, you don't want them to recombine. When they recombine, you lose the activity. So the higher the peak intensity there, it means the higher the recombination rate. So we see that the yellowish spectra there has the smallest peak, and this was when we added silver nanoparticles. This indicates that now our silver nanoparticles are acting as electron trappers, preventing them from going back to recombine with the holes, which means now we can maintain the photoactivity of the materials. We then further confirmed using EDX to check the elemental composition. And then we also used SEM to measure the sizes and also check the shapes of our nanoparticles. We then used TEM with a transmission electron microscope to check what kind of shapes do we have, which will also guide us as to how photoactive are they likely to be. And then we found that we have synthesized rod-shaped and pedal-like zinc oxide nanoparticles. We then calculated the diameters from the TEM images for comparison. And then we also calculated the pore size, the pore volume, and the surface areas. We did this because when you add surfactants in your synthesis method, you want to make sure that there's enough dispersion of the materials. So the more dispersed they are, the higher the surface area of your materials. You want to avoid agglomeration. So when we're happy with that, then we went on to conduct our photodegradation studies, where we prepared a solution of 10 ppm of the pollutants that we're using, and then we added 0.1 gram of the catalyst, and then we did the degradation. The last equation that you see there is the equation that we use to calculate the photodegradation efficiency. So we started our degradation studies with a dye which is called methylene orange. So figure A, we used zinc oxide as it is. Figure B, we used the one we synthesized with cyclodextrin, and figure C, we used the one that we synthesized using honey. So on image D there, you can see the color change in the solution, showing that there is some activity that is happening and the dye is being broken down. We then further went on to calculate the removal efficiency, which we found that we were able to remove 98.63% of methylene orange
using the cyclodextrin and the honey modified zinc oxide nanoparticles. And this was at the reaction rate of 20.59, the highest. So we further checked with another dye, and then we observed the same trend, that when we use honey and silver nanoparticles, we are then able to get a higher or a faster degradation rate of our dye, as we can see on the picture there, figure D. So when we are degrading rhodamine B, we obtain up to 99.98% removal efficiency at a reaction rate of 43.75%. This is a very fast reaction. So after being able to degrade this much in terms of dyes, we then went on to the more stubborn pollutants, which are micropollutants. And the figures A, B, and C, it's atrazine, which is mostly used as a herbicide. And then D, E, and F, that is bisphenol A, which is used as a plasticizer. And then the last one there is the chlorinated phenol. And then we observed that when it comes to atrazine, it's a little bit difficult to degrade them. I will show you the structure of atrazine and explain why is it difficult to degrade that. But then when we move to bisphenol A and go to 2.4 dichlorophenol, we obtained up to 100% degradation using the cyclodextrin and honey modified nanoparticles. So the structure that you see there is the chemical structure of atrazine. Atrazine has a lot of nitrogen atoms. These are highly high electronegative atoms, which makes it difficult to break the bonds, especially the double bonds there. Hence, we obtained the lower removal percentage of this material. We then calculated the reaction rate, and then we found that for the removal of dichlorophenol, we obtained the highest of 40.56, which is comparable to what we got with the dyes previously. So when you do photodegradation, the change in color that we observe in the solution does not necessarily imply that you are degrading. It could be a change in the shape or a bond direction, which may result in the change of color. So to confirm that we did degradation, then we run what we call total organic carbon. So the TOC measures the total organic carbons that you have in your solution. So when you degrade your dye, you convert it to your CO2 and water and other compounds, which then are released from the water as gases. So the drop in the concentration of that indicates that, no, you are actually breaking down the compound. So this was our confirmation, and then we also confirmed for the micropollutants. So the last part of this work was then to check if we can have all these compounds in one solution and be able to degrade them. So firstly, we combined the two dyes, and then by the degrees there in the peak intensity, we were able to see that there is some activity happening. And then we went on to combine one dye and one micropollutant. We made a similar observation. So we were happy, and then we went on to check with a mixture of micropollutants. We see that there is some activity, but compared to the other two, the activity is low. This is understandable because these micropollutants are more stubborn compared to organic dyes. We then went to check, to compare, when you degrade a dye alone and then you mix the dyes, what is the percentage that we're going to get? And then we see that we are getting the lowest removal of 63.44% and the highest of 98.28%. So we are comparing that when you mix BSA and DCP, you get 63% removal of both. And then when they are alone, you get 100%. So those are the comparison. The removal percentage is low, but our material are active, which is over 50%. It's good. And then we went on to do our TOC studies to confirm that indeed it was degradation. So in conclusion, we successfully managed to prepare a material using natural surfactants, which was photoactive. So to add the recommendations for policymakers, I divided them into 3. So the first one, we need to adopt green synthesis methods. Secondly, we need to support research on multi-component pollutant degradation, and then integrate advanced photocatalytic materials. We also look at water treatment. We are saying let's develop cost-effective water treatment technologies, and then enhance the water treatment infrastructure. And then lastly, encourage collaborations between research and institution and water treatment plants for upscaling, because often our research ends up in the lab. Now we need to move it to the next level. And then the last recommendation says, let's promote the use of sustainable natural resources, fund research on biogenetic catalysts, and also standardize green synthesis practices. These recommendations aim to support the development of more sustainable and effective approaches for environmental remediation, wastewater treatment, and the creation of catalysts using natural materials. With that being said, ladies and gentlemen, thank you very much.
Thank you very much for that interesting presentation. I like your recommendations at the end for policymakers. This was a very technical presentation, but I appreciated how at the beginning you simplified what the presentation was about when you said this is green chemistry to synthesize nanoparticles for water treatment. And so well done. We've run out of time. You used your whole allocation of 15 minutes. So thank you. Please help me thank Mandela again.
Thank you very much.
Okay, let's move on then to our next speaker, Ms. Shipalana. Did I get it right? Okay, thank you. We look forward to your presentation.
Hello, everyone. If you hear my voice shaking a little bit, it's because I'm a bit cold. I promise you're not making me nervous. But let's jump right into the research. What if I told you that the way, is it too far? Okay, is this better? Okay. No, this is okay. We can put it in. Is it not showing? Okay. Slideshow from the beginning. Is it better now? Okay, so what if I told you that the way we use our land is secretly draining our water resources? As someone who's lived in Guyana for most of her life, I know firsthand the effect that water scarcity has on local communities. Today, I'll be sharing my research on the connection between land use and land cover, as well as groundwater variability. So right here I have a map of the Greater Guyana local municipality in relation to South Africa, as well as the Mopani district. It is one of 5 local municipalities, and it is situated in the Limpopo province of South Africa. The municipality relies on both surface and groundwater. And lately it has been facing challenges regarding water scarcity due to population growth, agriculture, as well as the developing economy. The introduction to groundwater, land use and land cover change, as well as sustainable development. I have stated that South Africa faces significant water management challenges, and this was stated by the Department of Water Affairs and Forestry in 1997. The Greater Guyana local municipality has been facing water challenges for over 30 years, and this information I gathered during my honors research project as I focused on a similar study. I've obviously developed that now and I'm looking deeper into the issue. Groundwater is incredibly vital for sustainable development, and we need to consider how land use and land cover change can affect this phenomenon. I've listed here 4 things that we can use to integrate scientific research into sustainable practices. My first point is to enhance resource management, prevent resource depletion, climate change adaptation, as well as biodiversity conservation. Groundwater provides a reliable source of water for agriculture, drinking, and ecosystems, particularly in water-scarce regions like the Greater Guyana municipality. Land use and land cover changes alter natural recharge processes, and this affects communities and areas by increasing water demand and contributing to the depletion of vital water resources. My study's aim is to investigate the relationship between groundwater spatial variability and land use and land cover change using GIS and remote sensing. I've listed my objectives and I've highlighted the methods that I use to address each one. Assessing land use and land cover change between the years 2000 and 2022 provides enough time for analysis of how the changing environment and the changing land can affect water resources. Supervised classification is a way of teaching a computer to identify patterns in data and to classify it into categories. So this is how I did my assessment on Google Earth Engine. To analyze groundwater spatial variability, I used Kriging spatial interpolation, which is a technique used by taking data points and estimating values of the unknown points. Lastly, I determined the relationship by using multiple regression analysis. This analysis shows how multiple variables can affect a single outcome. So essentially, it's looking at changes in land use and land cover, such as change in vegetation growth or agriculture or urbanization, and how these things can be used as an input to determine a single outcome, which is the effect on groundwater. By accurately identifying these objectives, I looked into how to link this to sustainable development. Understanding groundwater variability helps inform better land management strategies and to ensure sustainable development in the following ways. Targeting resource allocation by identifying areas where declining groundwater levels, land management can prioritize regions that need more sustainable ways to deal with water scarcity. For example, recognizing groundwater depletion in Greater Guyana municipality could help implement solutions for the problem. Secondly, we need to have informed urban and agricultural planning because understanding groundwater trends can help to focus and develop better solutions for this problem and minimize environmental impact. And another example is how this land use and land cover change planning can protect the recharge zones in the Greater Guyana municipality. This is an overview of my methodology. I used Landsat imagery, which I directly downloaded from the Google Earth Engine platform, and used random forest classification to conduct the supervised classification required for the assessment. The groundwater analysis, as mentioned earlier, was done using Kriging spatial interpolation to map groundwater distribution over time. And another way I integrated data was using advanced spatial analysis techniques like the multiple regression model I mentioned earlier, and these will help to understand the relationship between groundwater and land use and land cover change. Google Earth Engine, for those of you who may not know, is a cloud-based geospatial data analysis platform. And it enables efficient processing and extraction of valuable insights, facilitating an LULC, which now I hope you've gathered is land use and land cover change, and it also allows to assess groundwater spatial variability also on the platform. This is still an ongoing study, so I would like all of you to consider the following hypotheses on the screen as I move forward into my presentation. Key findings that I've gathered so far are that urban growth, agricultural intensification, and vegetation change are the main causes of LULC. My focus was to determine how significant the change was affecting the groundwater availability and variability across the region, as well as the relationship between the changing environment as well as water resources, and how this implicates sustainable development. Urban areas have expanded in the area, and there are increasing numbers of declining groundwater levels in the region. Now, assessing the relationship between these two things draws a strong correlation, which I also discovered during my research. On the screen, I have screenshots of the code that I used to begin my analysis on Google Earth Engine, just so you can have a look on what it looks like on the platform and just the process that is required to start an assessment like this. There's one for 2000 and one for 2022. They look very similar because Google Earth Engine requires specific functions, and it is a very sensitive platform, which I've only begun to learn this year, honestly speaking, but it is very interesting and very efficient, and I recommend it for studies related to topics like this. Next, I have images of the Landsat imagery for the 2 different years that I extracted and clipped to my region of interest. And I've shown you the region of interest is the red on the side, and that's the Landsat imagery. I used Landsat 5 and Landsat 8 imagery for 2000 and 2022 respectively. It doesn't look very different on the screen, but I think it takes a bit of a deeper knowledge on GIS and being able to interpret. But if you focus clearly, I think you can see that the bottom one is a bit darker, which shows how the agriculture has definitely grown and how urbanization has increased. It is quite difficult to see, but there is a difference, and when I finish my research, I'll be mapping not directly with these images, but it will be interpreted differently on a GIS platform where I'll be using different colors so the images can be clear. These are screenshots from Google Earth Engine. The key findings that I have gathered while analyzing those images and during my process were these: that groundwater trends are significant, and the spatial variability, that is totally incorrect. Groundwater trends and impact of land use and land cover change. There's significant spatial variability within the area near agricultural and urban centers. There's also groundwater availability is also closely tied to these changes. I added a table here to show you that the classification of the groundwater. I assessed a sample of 400 boreholes, which I received the data from GRIP, which is a platform that is used to hold the groundwater data for Limpopo. I just forgot to add the link to show where you can access it. But the water can be classed from 0 to 4, and the higher the class of the groundwater means that the water is less, less safe to drink. Now, addressing the challenge, this research emphasizes the need for interdisciplinary approaches to topics such as this. I have a background in sociology, and so my research is heavily focused on communities, and I'm sure you've gathered that this is a very personal topic for me as I stand here representing my home. I think it is great to consider different perspectives and to look at things not only from a scientific perspective, but also from other angles. My recommendations are policy integration, sustainable agriculture, and urban planning. I believe that these recommendations align with Section 24 of the South African Constitution, which guarantees the right to an environment that is not harmful to health and well-being and ensures sustainable development through responsible resource management. By promoting these techniques, I believe that local governments can help maintain groundwater levels while still promoting and supporting agricultural activities or productivity that are essential for economic and social development. This is a recent case study from a bilharzia outbreak in Tanin, and there was an article by The Citizen that was posted on September 5th of 2024. I added this to my presentation so you can understand the implications of being negligent when it comes to water resources. A primary school was affected and now has a total of 142 cases reported of young children being affected by groundwater that wasn't correctly handled for quality and for safety. These are my interdisciplinary perspective recommendations as well. I stand for social science insight in topics of science that are related to large communities of people, as well as marginalized communities. Greater Giyani local municipality is a rural region, and as well as technological solutions. That's why I decided to integrate this into my research. Thank you.
Okay, thank you very much for that interesting presentation. I have questions, but I will find you during the break because you used up all your time. So let's move on. Let's move swiftly along to our final speaker, Ms. Ngwenya. We look forward to your presentation.
Thank you for the introduction. Can you hear me? Okay. Thank you for the introduction, and I'll take you through the work that I've done for my PhD. And just to give you a bit of background, so when you consider the human body, there are various processes and you get a lot of byproducts. And some of those byproducts in the body are what we call free radicals. Free radicals are essential in the body because they help maintain redox homeostasis, and they help in cell signaling, and they are also essential for immune responses. And this is important in immune responses because they help in acquired and in natural immunity. However, the problem lies when now there's overproduction of these free radicals. And this leads to chronic illnesses due to oxidative stress in the body. So basically, oxidative stress is a condition in which there's overproduction of free radicals, and then the body just goes through certain chronic illnesses. Some of the three major chronic illnesses that affect much of the world population include cardiovascular diseases. So about 17.9 million fatalities in the world, according to the World Health Organization, are due to cardiovascular diseases. And another major group is cancer. So according to the World Health Organization, in the midst of the pandemic in 2020, 10 million fatalities in the world were actually due to cancer. And according to Stats South Africa, in 2018, about 9.7% of the population succumbed to cancer-related diseases. And the last category is aging and brain disease. So brain disease basically is mostly Parkinson's and Alzheimer's, amongst others. But then these are the two major ones that affect the population. And about 26 million people in the world suffer from brain diseases. And age is a risk factor. Yeah. There are ways to mitigate the effects of free radicals, and this is through the use of antioxidants. So antioxidants are basically compounds that prevent the harmful effects of free radicals, and they prevent the overproduction of free radicals. I'm sure most of you here have seen the ad for Presidin on TV, YouTube, or something. That's an antioxidant, and that's what Presidin is. So this is where my research also comes in. I'm looking at coumarins. So coumarins are found in plants, and they are nutraceuticals, and they have broad pharmacological activity. What this basically means is they can be used to produce FDA-approved medicines, and they can also be used to develop novel synthetic coumarin compounds that can also be used as building blocks for FDA medications. However, the challenge that we face is the coumarins that have antioxidant properties in nature is quite low, and this is also very much environment dependent. And this poses a challenge because the production of antioxidants that can help the general population relies on the availability of those coumarins in nature. And this is where the research comes in, which is focusing on making synthetic coumarins that can be readily available for synthesis of other drugs. So now when we go into the synthesis of these coumarin compounds, I'm going to describe two methods of synthesis. So when you think about chemistry, there's what we call traditional chemistry, batch chemistry, which is basically a round bottom flask over a hot plate with a stirrer bar, your reagents, everything mixes, you get your product. However, this method, quite honestly, is it's outdated in a sense, and it also, number one, when you set up this type of reaction, when you are developing something that is going to be used in the long run, it becomes difficult to scale up into batch reactors because the conditions that you use on a pilot level are not the same conditions that you'll use at manufacturing level with big reactors. Therefore, with every development cycle, you have to change your conditions, sometimes change your reagents, and it becomes tedious. However, there's continuous flow chemistry, which is becoming widely available in the world. It's quite new in South Africa. Nelson Mandela was one of the pioneers of flow chemistry. UP has taken it up, UCT has taken it up, and this is basically a technology in which processes are done in a continuous stream instead of doing it in batches. So what you do, I'm going to play this video right here, and I want you to pay attention to the orange liquid. So these are pumps that you use to pump your reagents. So if you've got your starting materials of your reagents, you place them in syringes, and then you switch on your pumps, and the liquid flows through reactors. As you can see, the liquid flowing through the reactors, and then you can then collect your product for analysis and then determine what you have. Now, this technology is very essential because it's safer. It provides efficient mixing. The channels have grooves in them in which that facilitate efficient mixing. And when I say there's increased process safety, so when you look at the reactors, they are small and everything happens within the channels. And this allows you to control your reaction. You can control your flow rate, you can control the temperature of the reaction. And in chemistry, you get reactions that are highly exothermic, meaning that they can be explosive. So when you do reactors on this scale, in these reactors, you can control those type of things, sorry. And because now you're using a smaller system, you lower the cost in terms of like the pilot phase of your production, and you have shorter development cycles. And with these type of reactions, what you optimize at this scale, you can also transfer to kilo scale. So you don't have to re-optimize every time you have to scale up from micro scale to kilo scale. You can use the same reaction conditions, so you save time and costs. So for this project, our aim was to develop benign, high-yielding, and a clean approach towards the synthesis of coumarins, and also integrate this process of coumarins into microreactors. And with this method, what we aimed to do was to improve the production of coumarins such that they are readily available as building blocks for other end products that are on the market. So on this image here, I'm just showing you the complete setup for a flow chemistry system. It's quite a small system, as you can tell, and you basically get a pump, some syringes, you can have a thermometer in the reaction to monitor the temperature, and then your reaction plates are inserted into a beaker, and you can then optimize your reaction, whatever reaction conditions you want to optimize. It could be temperature, concentration, and everything else. So also bear in mind that this reaction setup is for lab scale. You can tailor it however you see fit for the type of production that you're looking at. So with this setup, this was the general scheme of the work that we did. We dissolved our starting materials in solvent, acetonitrile, and then we pumped it through reactors at a set temperature, and we collected our data for analysis. For this work, we aimed to develop a library of compounds. As I've mentioned, coumarins in nature are not readily available, they are low in concentration. So what we aim to do is to have a library of coumarins readily available as building blocks for other end products. And this table highlights the 3 major products that we focused on. There are other compounds. Our library has about 20 compounds that we synthesized for this work. So with this work, we realized that we could actually push the yield to almost 100% for some other products. And using one method, we could apply it over a range of starting materials to get various products. And this work is important because flow chemistry is a great technology to adopt for API manufacturing, which is active ingredients. Just to give you a bit of context, South Africa, as it stands right now, has 265 manufacturers of API manufacturers, and over 50% exclusively supply the private sector. Oh, APIs are active pharmaceutical ingredients, right? So the country currently has 265 manufacturers of active pharmaceutical ingredients. Over 50% exclusively supply the private sector. 10% supplies the public sector, and the remaining percentage supplies both sectors. Now, this is a very concerning thing because the government then has to rely on imports of active pharmaceutical ingredients for medication. And as it stands, the government spends about 8 to 9% of the country's GDP on importing drugs that can be used for medicines, and that's about 44 billion rand. And I'm sure we all saw this with the COVID pandemic when the government had to import vaccines for vaccinating the population. And This graph just shows you like the major countries that the South African government imports from. India and China are the leading exporters of active pharmaceutical ingredients to South Africa. And then you have your US, your France, Germany, and Ireland. So my recommendations are basically, the government should be able to identify and invest in key and emerging technologies. As we can see that flow chemistry, we had it in Mandela for a while before it was taken up by other universities like your UP and your UCT. And I think it's important for the identification of these technologies that actually advance the community and the country as a whole. And there should be collaboration between various sectors, academia, industry, and the government to ensure that there's accelerated innovation in commercialization of new technologies. And also there should be technology transfer IP policies that promote collaboration between various sectors. And also we need to develop shared infrastructures between these various sectors. And also we need to ensure that these new technologies contribute to sustainability goals. So my research particularly focuses on SDG number 3, which is good health for all. If we can provide good health for the population of South Africa, then we are able to meet the SDG goal number 3. Thank you for your attention.
For that presentation, we have 2 minutes for questions. Do we have any questions from the audience?
Thank you for a wonderful talk and such an interesting, presentation. My question basically is on the route that the system itself, the methodology itself. I've seen you have managed to obtain about 100% product of your interest. Does that say that along your your synthesis route, however you use all those reagents, you have less of byproduct, or you don't have byproduct at all?
That's actually a good question. So one of the major components that flow chemistry aims to address is selectivity. So to explain it properly, when you mix your reagents in a round bottom flask, there isn't efficient mixing. But whereas when you mix in the reactor channels, so I'm not sure if you can see from this video right here, when it gets to the second plate, the liquid doesn't flow in a straight line. It flows into grooves. So those grooves allow for efficient mixing of your product, and that is what pushes selectivity towards your desired product. So what we do to ensure that we actually get the yield that we get, in this case the 100%, you have to alter other reaction parameters. Could be temperature, it could be the concentration of your reagents that you're using, it could be the flow rate, how fast or slow are you pumping your reagents into the channels. And yeah, so when you do flow chemistry, there's a lot of optimization. You play around with stuff. So that's what leads to good selectivity.
Please help me thank Ms. Ngwenya for her presentation. So thank you very much. I think in this session you have taken us on a great journey from looking at addressing diseases in maize to green chemistry in the synthesis of nanoparticles for water treatment. We have looked at sustainable practices in groundwater use. And now finally, we've looked at the use of flow chemistry in possible production of active pharmaceutical ingredients. What a rich series of presentations. You know, often they say the future is bright because of the youth. This was an example of something like that. And so please give all the presenters a round of applause. I also hope that through the presentations that were shared this afternoon, that you will have many questions to engage with the speakers about during the gala dinner. We can't do it now because we've run out of time, but find each other during the gala dinner and continue the conversation. These are really great conversations on science and addressing societal challenges. Thank you for attending this session. We have a break now, and we will see all of you back at the gala dinner. Thank you.
endings lie. I love the drama 'cause I never know what the endings lie. Please make me small so I don't feel important, so I don't waste it all and be wrong about what I want. I know I'm nothing, but my mind keeps lying. Speck of dust, blink your eyes, and we're gone. So let's live. Let's live. You know what I mean? That's right. That's it. Let's live. Let's live. So beautiful, but I'm blind now. Oh, no. Let's wake up now and not miss it anymore. Born again, better late than never. I'm not done. Wish me luck on my new trips around the sun. Don't let it get you. 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 Let's live. Let's live. Let's live. Let's live. Again, I close my eyes. I close my eyes. Play back, repeating the same old night. back alone. Come back. Ready to live steady now, but no one wanna talk. Depending on us all, cause your mission didn't work out. You didn't know time would slip this fast. Now it's too late. Now it's too late. Now it's too It's always greener on the other side. Always greener on the other side. It's always greener on the other side. Always greener on the other side. Oh, yeah. You open your eyes, thinking for what you sure.